Good morning, good afternoon, and good evening, or whatever time of day it is for all of you around the globe who are participating, and welcome to our second day of the third International Remote Conference, Science and Society. It was a big success, and hopefully today will be the same. I just wanted to let you know that this year we have to date about close to 540 registrants for the conference from approximately 52 countries. And just to give you a flavor of where these registrants are coming from, we have Mozambique, Senegal, Gambia, Philippines, Poland, France, Netherlands, the UK, Italy, Finland, Canada, the US, Lithuania, Guinea-Bissau, Kenya, Nigeria. In South America, we have Bolivia, Argentina, and Brazil on board, Honduras and Mexico, Australia, China, and I could go on and on and on. So I'm delighted that there are so many participants. And what I'd like to suggest, and I'll reinforce this at the end of the conference, is that for those of you in various countries who'd like to start your own Beyond Sciences initiatives, BSI chapters, we're here to support you and can get you up and running and get your website organized. And all you have to do is email us at beyondsciencesinitiative@gmail.com, and we'll provide you with the tools and the support to make this happen. For you to be able to network, interact with scholars in your country, in neighboring countries around the world, peer-to-peer contact, perhaps talk about science, engage in outreach activities. As I said, we're here to support you. I want to thank again our sponsors. That's Trinity College at the University of Toronto, the Department of Immunology at the University of Toronto, BioLegend, BMO, and Janice Wright. I'd also like to thank all those at the Toronto BSI chapter, the BSI chapter in Kenya, the two various chapters, and in India for also supporting us. And obviously for the group here in Toronto and Canada who've made this happen at the hub of the Activity Center. So without any further ado, I'd like to invite Payam Zarim to now introduce our first keynote speaker of the day. Over to Payam.
Good day, everyone, and thank you for joining us. It is my pleasure to introduce Professor Bruno Silva-Santos. Bruno is a professor of immunology and vice dean of the Institute of Military Medicine in Lisbon, Portugal. Bruno trained as an immunologist at the Cancer Research UK at King's College in London under the mentorship of Dr. Adrian Hayday. As an independent group leader, Bruno has been funded by many prestigious grant agencies, including the European Research Council and the European Molecular Biology Organization. His lab is focused on a variety of topics, including the activation, development, and function of gamma delta T cells, ranging from everything in what happens in the thymus to what happens in peripheral tissues. Some of his seminal papers include a paper in Nature Immunology, which was the first to discern between IL-17 and interferon gamma producing cells in a really fundamental way by showing that CD27 positive gamma delta T cells are interferon gamma producing, whereas the CD27 low cells are IL-17 producers. However, the some of his research stops. He also has quite a lot of great papers focusing on cancer and immunotherapy. And this is where his talk will take us today, to seeing how we can translate some of the findings in the lab into real-world advances. So without further ado, I invite Bruno to start his talk, and I hope you enjoy our first keynote of the day. All right. Bruno, can you hear us?
Yes, I can.
Okay, wonderful. We have you, we have vision, we have a good voice. Okay, best of luck.
Yes. Good day, everyone. Thank you very much, Eleanor and Payam and Ashton. together and really for inviting me to talk about our work on cancer immunotherapy. I'll actually start with an introduction to the field and what's cancer immunotherapy nowadays and where we intend to go by developing a new type of immunotherapy based on a cell type that we are very focused in the lab that Payam just mentioned, the so-called gamma delta T cells. So I'll start by really saying that today, cancer immunotherapy is seen as a change in the paradigm of cancer treatment because unlike other therapies that, as you can see, target the tumor, both chemotherapy, radiotherapy, or the most developed targeted therapies, all of those have the tumor as the target. In the case of immunotherapy, we're trying to actually use the immune system of the host to do the job. And so what we need is to activate the immune system, and there are several ways to do that. And the immune system itself and its components, in particular its T cells, will destroy the tumor cells. And as you can see on this timeline, this is really the new kid on the block, because whereas other therapies have been around for many years, immunotherapy, as we see it nowadays, it's only around since the beginning of this decade. There have been, however, other types of immunotherapy tried, but not really reaching the clinic in the global way that we'll mention today. And so immunologists have been following cancer immunology and immunotherapy as an objective for over 1 century. And finally, this decade, we have been able, we as a community, to produce the goods. And these have come in the way of all of these approvals by the FDA or the EMA in Europe of drugs that really, these are biologicals or cellular therapies that by definition are immunotherapies. What you have here are 3 antibodies that are immunomodulatory. So they really, what they do is to activate the T cells of the patient to fight its own cancer. And the final one, approved just a few months ago, is a cellular therapy that I'll mention towards the middle of my talk. And so what we have here is a list of tumor types where immunotherapy is now producing the goods. So it's no longer just melanoma, which is the easiest tumor type to really target using immunotherapy because the cells are particularly immunogenic, but also non-small cell lung cancer, a very difficult cancer to treat until very recently, and that now, for at least a third of the patients, immunotherapy is really the option. Then we have bladder carcinoma, renal cell carcinoma, and also some hematological malignancies like Hodgkin lymphoma and some types of leukemia, in particular acute lymphoblastic leukemia. And so there are several types of cancers that for which immunotherapies are already a reality, and we want to increase this list, obviously, and I will tell you a bit about these efforts. In all cases, we are trying to use so-called anti-tumor lymphocytes, so white blood cells that are endowed with properties to fight the tumor in one way or another. And in particular, we're thinking about killing, since we have to destroy the tumor cells one way or another. And therefore, it's very important to realize that within this set of anti-tumor lymphocytes, we really have T and NK cells endowed with the capacity to kill. These are the only cells in your body that produce venoms, poisons, that are able to kill other cells. And this is through a series of reactions using these kind of enzymes. Perforin, which creates a pore in the target cell, the tumor cell in this case, and granzymes that will penetrate the tumor cell and induce apoptosis by activating the intracellular cascade system. And so granzymes and perforins that are expressed upon activation of T and NK cells, these really endow them with the capacity to kill tumor cells. And this distinguishes T and NK cells from every other cell of your body. And we are going to concentrate on T cells. And as you can see, T cells really create very close contacts with cancer cells. And as you can see, this T cell here is establishing the so-called synapse, not the neurological one, but in this case, an immune synapse with a cancer cell, and is trying to detect on the cell surface of the cancer cell antigens that will allow the T cell to recognize this cancer cell as different from a healthy cell. And what type of antigens can a T cell recognize? Well, for the prototypic T cell that kills tumor cells, the so-called CD8 T cells or cytotoxic T cells, they have a T cell receptor, and this T cell receptor interacts with the MHC class 1 that is expressed on all cells of our body except red blood cells. And so all cells of our body, including cancer cells, at least at the beginning, express this MHC class 1. And this is a platform on which small peptides derived from intracellular proteins are exposed to the T cells. So the T cells can screen these peptides and really decide whether they will mount a response against the cell if these peptides are strange, aberrant, something that will allow the T cell to recognize that different from self, or will tolerate these peptides in the case of self-protein. This is what we want and how our system is tuned. Of course, we know this can go wrong, for instance, in autoimmune diseases, but that will not be the topic of this talk. So what kind of peptides can be there on the surface of a tumor cell that will allow the T cell to recognize it? Well, the easiest peptides to allow such a recognition are obviously peptides derived from transformed proteins, mutated proteins. So during cellular transformation, as we all know, tumor cells acquire mutations. And so if the mutated proteins generate peptides that are exposed on the cell surface, this will allow the T cells to recognize those cells as being different from healthy cells that do not bear these mutations. And so since the beginning of a tumor development, there is this constant crosstalk between immune cells and tumor cells. And at the beginning, this is a very famous theory in cancer immunotherapy or immunology called the cancer immunoediting theory by Rob Schreiber, where it was postulated that there are three phases of this interaction between tumors and the immune system. As you see, the first phase is elimination, where the immune system is winning the battle. Then there is an equilibrium in which there is a point that will eventually be overtaken by the tumor, and the tumor will dominate upon the so-called escape. So when we diagnose, when a cancer is diagnosed in the clinic, we are invariably at this stage, the escape stage. The tumor is already there. So it has avoided all the killing mechanisms that I talked about from T and NK cells and has established a mass. So obviously, the only way to now tackle this tumor is to really go back and try to revert this process back to the elimination phase. And so how does the tumor get to the escape? Well, one of the major breakthroughs in the last decade, and this allowed these new immunotherapies to really come through and be approved, was work from the end of the 90s by Jim Allison, Onjo, and other pioneers that identified molecular breaks on the cell surface of T cells. And so these two receptors that you see here in red, CTLA4 and PD-1, these are really inhibitory receptors, so they are breaks to the activity of T cells. These are critical breaks because if we don't have them, for instance, if we generate mice that are knockouts for CTLA4 and PD-1, these mice kill themselves within weeks after being born of fulminant autoimmunity. So these are the breaks that prevent self-reactive T cells that we all have from destroying our tissues. However, the tumor takes advantage of these breaks to actually trigger them and therefore avoid that the T cells will mount a response against the tumor cells. And as you can see here, this is in the case of PD-1. When PD-1 is ligated by its ligand that is expressed on the tumor cell, PD-L1 in particular, this provides an inhibitory signal to the T cell that will really stop all the T cell activation that the T cell receptor, as I mentioned before, is trying to set up. So the T cell receptor recognizes the antigens and tries to activate the T cell, but if PD-1 and CTLA4 are themselves ligated, this inhibitory signal will actually overcome the activatory signal and will block the activity of this T cell. So as you can imagine, a logical way to tackle this process is really to block these inhibitory receptors, these breaks, in order to revert this process so the T cells that are now stopped by the tumor can reactivate. And so in immunotherapy nowadays, we really have two main strategies. The first of which that generated these first three antibodies I mentioned as being already in the clinic, anti-CTLA4, anti-PD-1, and anti-PD-L1. All of these tackle the so-called checkpoints of the immune system. So these checkpoints try to prevent autoimmunity. The tumor takes advantage of that. So the therapy consists in using antibodies that prevents these breaks from being triggered. So the tumor, even if it expresses all kinds of ligands, can no longer trigger the break, and therefore the T cell is free to exert its activity. So this is strategy one. And strategy number two, that we'll mention afterwards, is cellular therapies. And it is in this context that we develop our own research, trying to use cells from the patient to actually kill the tumor. So this means taking blood from the patient, manipulating this blood in the lab, and sending millions of chosen T cells back into the patient to do the job. And so just to show you how these the antibodies have made such a difference, this is non-small cell lung cancer. And for 30% of the patients that express high levels of the ligand of PD-1, PD-L1, this means the tumor cells have high expression of this break and will trigger this inhibitory mechanism on the T cells. For these patients, you can see this massive difference. This is progression-free survival of patients treated with anti-PD-1, this purple line, or with the conventional chemotherapy, which was docetaxel, which was the only thing available until recently. You see a terrible outcome for all these patients treated with chemotherapy. Simply, it was not working. Whereas now, for 50% of the patients, 60% or 50% at 1 year, rather than 15% with chemotherapy, we see that there is no progression of the disease. And now these responses are being maintained for at least 1/3 of the patients in longer terms. And a lot of these trials are still ongoing because, as you can see, a lot of this research is very recent. This paper is less than 2 years old. And so we are seeing durable responses in a large proportion of patients in some types of cancer using these antibodies. And the cancers that are producing the goods are actually these ones: melanoma, lung cancer, bladder cancer, etc., that are on this edge of this graph. And this is an important graph because it shows you the amount of somatic mutations per tumor type. And so this means that tumors that are very aberrant, that are very extreme, very highly mutated, melanoma because of UV radiation, lung cancer because of tobacco smoke, these are very easy to recognize by the immune system if you remove the break. There are enough mutations to recognize these antigens. The biggest problem for this kind of strategy is the other extreme of this graph, which are mostly hematological cancers. A lot of leukemias are there, where you can see the mutational load is around 1/10 or 1/100 of that of melanoma and lung cancer. So these are poorly mutated cancers for which releasing the brakes is typically not enough because the cells are not easy to recognize by the T cells that are now reactivated. And so for these tumors, adoptive cell therapy really seems to be the most suitable strategy, where we use T cells focused on particular antigens of those low mutated tumors to actually tackle those tumors. And so adoptive cell therapy needs to get the white blood cells, the T cells in particular, we're thinking about selecting them, expanding them to millions or billions in vitro, and re-injecting them back in the patient. And the therapy that has already been approved is the so-called CAR T cells. So CAR stands for chimeric antigen receptors, and these are called chimeric because these molecules are actually a mixture of a B-cell derived molecule, which is an antibody, with molecules typically seen on T cells. So as you can see in this slide, the domains of this molecule, which is engineered and put on the surface of the T cell. So here you have the T cell, which is born with a T cell receptor, but by genetic engineering, it acquires now a CAR. And this CAR, chimeric antigen receptor, is this very exquisite molecule which has an antibody-like recognition domain on the outer surface, and then intracellularly, it has the domains required to activate the T cell. So this allows to recognize a tumor with an antibody-like motif. What's the advantage? Well, antibodies recognize proteins face-to-face, so they don't need MHC presentation, and MHC downregulation is one of the major immune evasion mechanisms that tumors employ. So when tumors try to escape the immune system by taking MHC class 1 from the surface, now by having a CAR that does not rely on MHC, because it has an antibody domain on this molecule that recognizes a given tumor antigen, we can have T cells to recognize every cell in the body that has this antigen on the surface. Of course, the big challenge now is to have a relevant tumor antigen that is specific enough so that these cells will not target healthy cells, and that is broadly expressed on tumor cells enough that all tumor cells of that given tumor are targeted, so we can actually eradicate the tumor. And so there is still very few tumors for which we have a suitable antigen. And this is the biggest limitation of CAR T cells. So the only CAR T cells that have been approved are the ones that recognize these molecules, CD19, which is an antigen expressed on B cells, and therefore suitable for B cell leukemias. And there is a very promising molecule for another B cell derived cell, which is a plasma cell, that is the transformed cell in the multiple myeloma, and BCMA CARs are also producing very interesting results in the clinic. So CARs are very complicated to generate, as you can imagine, because you have to take the antibody domain that comes from a B cell receptor and fuse it with all these domains of the T cell for activating the T cell, put it into a lentiviral vector, and transduce your T cells, and then we inject them back. So it's definitely not an easy thing to do, but it's really producing amazing results in B cell malignancies. So for B cells for which CD19 is a suitable antigen, we're getting amazing responses in these acute lymphoblastic leukemia patients. These are data that are less than 2 years old, but that led very soon after this, in 1 year, to the FDA approval of CAR T cells recognizing CD19. Now, there is a problem, though, with CAR T cells, as I told you, because there is a huge, not only we have to identify a very good antigen, and CD19, you should realize, it's not a tumor antigen, it's a B cell antigen. So we wipe out all B cells by using CD19 CAR T cells. It just so happens that we can live without B cells. We provide antibody, intravenous, IG, immunoglobulin, and these patients are protected from infections from the antibody they get. But most importantly, they are tumor-free because all B cells have been eradicated. Of course, this is a strategy we cannot use for an antigen in the lung, for instance, because we cannot destroy our lung overall. And therefore, we need to get better antigens for CAR T cells to be more broadly applied in cancer immunotherapy. Another limitation is by having such a pressure on a given antigen, there will be selection for so-called escape variants. A few cells of the tumor that do not express that antigen will have a massive advantage. And so they can relapse and really reconstitute the tumor after the first mass has been eradicated. And we're already seeing this even with CD19 CAR T cells, where a new tumor comes up that is CD19 negative. So CARs have their limitations, and this is why we are trying to develop new immunotherapies for cancer. And our immunotherapy is called DOT cells, for delta 1 T cells. This is a gamma delta cell that we are trying to make NK-like, natural killer-like. Both NK cells and NKs in general don't recognize MHC and peptides, so they, again, are not problematic to use against tumors that have downregulated MHC class 1. And we developed these DOT cells by differentiating the regular gamma delta cells that are present in the body of the patient or ourselves into cells that have a different phenotype. Essentially, as you can see here, these are regular gamma delta cells, and suddenly our DOT cells have a high expression of this receptor, NKp30. As the name says it, this is a natural killer receptor that we are now expressing on the surface of gamma delta cells. Normal gamma delta cells never express this molecule that was supposed to be NK cell specific. But we found a way with a patented protocol to induce the expression of this molecule. And so this NKp30 is now sitting on the surface of the DOT cell, and this recognizes an antigen called B7H6 that thankfully is not expressed on healthy tissues, but is highly expressed on tumor cells. So this allows a new recognition mechanism for dot cells to see tumors through BS7H6 expression. And we think NKp30 is very useful because, as you can see in these experiments, when we trigger NKp30, we can render a tumor that was resistant to gamma delta cells. This is killing of the tumor cells by gamma delta cells. Initially, very little killing, but if we provide NKp30 activation, we now get a very strong killing. And more importantly, we also really double the amount of interferon gamma these cells produce. Interferon gamma is a very important anti-tumor cytokine. So we try to really scale up and produce enough of these dot cells for adoptive cellular immunotherapy. A problem we had is that these gamma delta cells we're trying to use are not very abundant to begin with. This is a blood sample of a patient, and as you can see, only 0.3% of the white blood cells are these B delta 1 that will generate the dot cells. So how can we expand them and make them strong anti-tumor effectors with clinical grade reagents for the clinic? How can we do this? And to set up this, my first PhD student in the lab, Daniel Correia, convinced me that we should start a company. And so he went into an entrepreneurship course to get all the knowledge required and to get partners to get a company started. And this company was started 5 years ago, and it's called Lymphact. And within Lymphact, we've been able to really optimize the process to expand these dot cells. And so we tested, Danielle did, tested 2,488 different combinations of media using all kinds of molecules, including cytokines and antibodies, to activate our gamma delta cells to produce dot cells in large numbers. And you can see here, these are different donors. We got to a very robust process that allows a huge increase in the expansion of these cells. From each gamma delta cell, B delta 1 cell we take from the body, we can generate up to 40,000 cells in life. So we now can generate a huge army of dot cells to re-inject back into the patient and have a chance really to kill the tumor. And so besides NKp30 that I mentioned, a series of other NK receptors, all of them used to kill NKp44, NKG2D, DNAM1, all of these are upregulated. What you see in gray is the cells we have in our blood, and in the black line is the dot cell product. And you see this shift in all of these molecules, meaning that all these receptors are now burning hot on the surface of dot cells, and all of them will allow them to kill. And they do kill. They kill a lot of different tumors, as you can see here. In gray, you have the control without dot cells, and in black, the tumor cells incubated with dot cells for just 3 hours. And for all these types of tumors, from hematological to solid cancers, you can see over 80% of killing of tumor cells within 3 hours by our dot cells. So they're extremely effective at killing a variety of tumor types. And we have so far concentrated on leukemias, but we are extending our studies to all kinds of solid tumors like ovarian cancer and others. Just to show you some data on leukemias, these are a series of leukemia AML, acute myeloid leukemia cell lines, and you can see how from 5 different donors we generated dot cells. In all cases, we can very extensively kill the tumor cells using dot cells. So all these leukemia cell lines, so only one was partially resistant and took longer than 3 hours to kill, but all the others in 3 hours are essentially killed. And even cells that have resisted chemotherapy and that are really representative of a relapse, they can still be killed by dot cells. So we really think dot cells are very, very interesting to use to kill these leukemia cells. And this is in vitro. These are assays in vitro. Of course, we wanted to validate this in vivo, just to show that in vitro, these cells kill the leukemias, but not healthy white blood cells. So when we co-incubate dot cells with white blood cells, there's no killing, whereas these are primary samples from 3 CLL patients, and here we have 2 other leukemia patients. In all cases of leukemia, the cells are targeted by the dot cells, and healthy cells are not. And so we wanted to go in vivo, so we established a tumor model in the mouse, and we saw that dot cells were able to home to the tumor, and within the tumor, these cells really remained. You see them here by fact. They were present in the tumor mass that was created, but they also were able to penetrate a series of different tissues, like the liver of the mouse, the lung, the spleen, and the bone marrow. And so they have these tropisms for tissues that we realized to be extremely interesting because it will provide a protection of all these tissues against tumor spreading, as I'm going to show you. Just to show you that T cells that infiltrate all these tissues, even 2 months, we are looking at them 2 months after administration in the mouse, they are still proliferating, which is amazing. Cells are so activated, they keep proliferating. This is a marker for cell activation and proliferation, Ki67, and they continue to produce interferon gamma, which is a very potent anti-tumor cytokine, as I mentioned. They do not produce IL-17, which would actually be counterproductive in this case. And these cells, although they infiltrate all these tissues, they don't create massive inflammation in the tissues, so they have a good safety profile. This is in the tissues by looking by histology and also looking here at blood biochemistry for renal and kidney function. All the enzymes of the kidney and the liver are normal between untreated animals and dot cell treated animals, showing that dot cells in large numbers in the mouse do not attack these tissues and do not produce effects that are worth mentioning. Of course, we have to see the side effect in patients, but so far as mice go, this therapy has a very good safety profile. What about efficacy? And this has been the major accomplishment of this therapy. So what we see here is the dissemination of a leukemia, and in the control group, you can see the tumor cells, which are in this case. This is human CD19. For human CD19 T cells, in this case, the leukemia cells, spreading from the tumor site to the liver, spleen, bone marrow. The bone marrow is full of leukemia cells, as you would imagine, is the ideal organ for the leukemia, and the lung. And these are the treated animals. These are the control animals. When we treat these animals with dot cells, a different group of animals, obviously, you can see these cells are essentially gone. So compare these two groups, the amount of tumor cells in the control and the rarity of these tumor cells in the treated animals. And this is by histology in groups of 6 animals. In black means the tumor is present, in white the tumor is absent. And you can see dot cell treated animals are essentially tumor free. Only in one animal we could see a bit of tumor mass in the spleen. In all tissues and for all other animals, these were tumor free, whereas the majority of control animals had tumors in all these organs. So we are very excited with this data. This is with chronic lymphocytic leukemia, and we have extended this data to another leukemia, acute myeloid leukemia, very, very aggressive leukemia. And you see dot cells in red can really reduce the tumor load in the blood and in the liver. So you have non-treated animals and dot treated. You have a massive decrease in tumor load in the target tissues, in the circulation of these animals, and this prolongs the life of the animal. So with all this data, we really want to take dot cells from mice to humans, obviously, and we are now in a very exciting stage of our company where we are trying to merge with a foreign company that will have the means to take us to clinical trials. We want to purify delta-1 cells from the patients, create their own dot cells in billions, and re-inject them back to the patient and see what is the prospect of us being able to provide a new immunotherapy, a new solution for this patient. So I'd just like to finish, before I take some questions, by acknowledging the team that has been involved in this work and our funders. Thank you very much. And I'll take any questions.
What's it? Thank you for the. Okay, so I'm going to start by a few questions for myself. We already have a few questions in the chat, and I encourage the participants to keep asking questions. So my first question is compared to some CD8 T cells that are being prepared for immunotherapy. Is there any difference with using gamma delta cells as kind of the cells in the therapy, and what could be the advantage?
Yes, so we think basically that our cells, dot cells, have a broader recognition mechanism in the sense that they use their T cell receptor that they are born with, but also this set of NK receptors that CD8 cells don't express. The only one they express is NKG2D. So CD8s are very T cell-like. So they do not have these NK properties that we are endowing our T cells to acquire. So essentially, we see dot cells as a hybrid of T and NKs. And you know, NKs are critical. If you have an NK deficient mouse, tumors develop like crazy. But unfortunately, we have not been able to use NK cell therapy efficiently in the clinic because NK cells are very difficult to manipulate. Our T cells, our gamma delta cells, are easier to manipulate. They expand more. They have a lot of T cell characteristics, but we add on top of these NK cell characteristics that allow them to see tumors in an innate fashion, in a quicker way, and in a broad way, which we think is an advantage to the CD8s that have a very antigen-specific way of looking at tumors. And we know this can be good, but we also know new tumors find a way of evading the CD8 strategy.
Thank you for the great answer. My other question is that I suppose what I found very impressive is that you can manipulate and have these cells proliferate 40,000 fold. So of course, after so many rounds of proliferation, I'm curious if you've done any RNA-seq to see globally how the gene expression changes between what you started out with and basically what you're proposing to put into the patient. What is changing in the cells?
That's an excellent question, Payan. So what we've done is we have not done our NGS or RNA-seq. These studies are ongoing. We started by cell surface typing. So we did 326 cell surface markers to really get a full repertoire of cell surface receptors. And we were very surprised that exhaustion markers like PD-1 itself was not expressed on these cells. Only 20% of the cells express PD-1, a minority of these cells. And you'd expect these cells, as you're implying, to go through all these divisions to be chronically activated, possibly to enter exhaustion. But both by telomere length that we measure, and they don't have shorter telomeres, and by the expression of these exhaustion markers, which they don't express, these cells seem to be very active still. And actually, that's what we see in vivo, because after these 3 weeks in vitro, we put them in mice, and 2 months later, they are still proliferating in all these tissues of the mouse. So it's really amazing. We are actually concerned, or on the other hand, we are concerned how to stop these cells. They are so activated. How can we stop them? What are the natural mechanisms to inhibit these cells? This is what we are not sure. So we are more concerned with stopping them than to keep them going, because they still have their own means, which is incredible, to keep going. And for you that are a gamma delta cell expert, I can tell you this is very specific to the V delta 1 subtype of human gamma deltas, because the V delta 2, which are the dominant ones in the blood, they cannot stand this chronic activation. They will die. So there's something intrinsic to these cells, delta 1. They love to be activated. They keep on going. And this allows us to really have a massive advantage for cancer therapy, where we just want cells not to stop until the job.
That's really fascinating. Thank you again for this great answer. So I'm going to go to the floor now and ask you some questions from our participants. So first we have Gitana from Lithuania, who asks, what are some of the side effects of immunotherapy? And for example, what should a physiotherapist who is involved in physical modalities and the kind of other non-drug cancer treatments be aware of as far as immunotherapy goes?
Yes, so with the antibody-based therapy, the major side effects are clearly autoimmunity. So by removing the brakes on our T cells, our self-reactive cells really are activated, and we see, for instance, in melanoma, a lot of vitiligo. A third of the patients develop vitiligo, an autoimmune disease of the skin, and there is attacks of the kidney, of the liver, and so autoimmunity is the thing that really worries doctors when they use the antibody. In terms of the cellular therapies, the major problem, so-called cytokine release syndrome, because we give so many millions of cells to the patient in one go that there is a huge activation of these cells. They produce a lot of cytokines, including interferon gamma, TNF alpha. This can lead to fevers, to myalgias, to these kind of effects that are very acute, but they have to be followed very, very carefully, because if there is, for instance, a disruption of the blood-brain barrier, this can lead to death. So there have been a couple of cases of CAR T cells therapies that led to death because of neural edema in brain. So this has to be followed very carefully by the doctors. Essentially, with cellular therapies, the moments after injection within the next few days, and in the antibodies during the few months after injection for autoimmunity, any kind of symptom. If those occur, the advantage of autoimmunity, we know how to tackle it. So there are drugs that paradoxically will tune down our immune system, and they can tackle the autoimmunity. And what's amazing is that it seems that when you go and tackle the autoimmunity, so you stop the anti-cancer treatment, you tackle the side effects, and then you revert to anti-cancer treatment when the autoimmunity is controlled, you do not lose efficacy. So it's amazing that you can go into cycles of kill tumor, control autoimmunity, kill tumor, control autoimmunity without losing efficacy. And this has been a major surprise in the clinic.
That's a great answer once again. So to follow up on that with two things, one a question from the audience and one a question for myself. This effect that you see as far as autoimmunity goes, do you know how much of it is from the cells that you're providing yourself and how much of it is from other cells being activated? And if it's the cells that are being provided, are there kind of suicide receptors that you can have that can turn off, basically if you give a drug and you turn off what the cell is doing and turn it back on? Is this something that's being tested?
It's a very good question, and it is indeed. Actually, for CAR T cells, this is seen as the next generation, to have suicidal genes easily activated by a small drug that can be injected in the patient to really wipe out these cells if the problem starts occurring. So for CAR T cells, definitely. For our cells, I have to say, we do not anticipate that. So, gamma delta cells are not MHC restricted, so we are not foreseeing any problem in terms of autoimmunity per se. We are concerned with cytokine release, and so we can destroy the T cells by using a general antibody against T cells if necessary, but we are not currently transducing the cells with any suicidal gene. We might still end up there if we feel that there is any safety issues to be addressed. We shall see. We'll take it easy. We'll do a dose escalation assay in humans, of course, and if all goes well, the maximum tolerated dose will then be used for efficacy trials in phase 2. And we are hopeful that also based on the mouse and on the literature on these cells in humans, our starting cells, the B delta 1, that the side effect will be controlled, but we shall see.
Thank you. We have quite a few questions. In the interest of time, I'm going to just merge one more question from Finland with one from Toronto. So from Finland, we have a question that asks, would this treatment also protect the patients against opportunistic infections that are associated with cancer, such as by other pathogens or bacteria? And from Toronto, we have a question that says, I find it remarkable that your dot cells are killing tumorigenic cells with no off-target killing of healthy cells. Although your dot cells are fundamentally gamma delta T cells, is there a possibility that they're recognizing a tumor-specific antigen? This sounds like it's almost directly from JC.
Yeah. So very good questions, both of them. I'll start with the second one. Yes, there is the possibility. We are trying to really discriminate between how much this recognition is dependent on T cell receptor or on the NK receptors that we induce. Because we see better killing from our dot cells compared with their mother B delta 1 cells, we definitely know that the NK cell receptors are doing a job. And actually, if we trigger those receptors, as I showed you, we can induce killing of a tumor that is very resistant if just relying on the T cell receptor. But for other tumors, the T cell receptor may be very well capable of recognizing tumor antigens and be doing a job. So we're trying to dissect these two components. We're trying to use CRISPR-Cas9 technology to manipulate these receptors or their ligands on the tumor cells and to really discriminate these two components. For now, we don't have enough information. And of course, in general, for gamma delta cells, the identity of these ligands is the question in the field, so the holy grail. And we really want, in the context of the new stage of our company, to have a line of research just to identify TCR ligands that are relevant for our dot cells and the NK receptor ligands as well. So to have the two lines of research side by side, but it's still too early to know. The first question, what was the first question again, Fayan?
If this affects opportunistic infections, yeah.
So that's a great question. Actually, our dot cells are there. We have a paper on that. They are very, very good at trying to generate antiviral responses. So the V delta 1 cells, from which we generate dot cells, they are essentially antiviral. So they are very poor against bacteria. They are very good against viruses. And actually, the best correlation against CMV reactivation, when you mention in cancer patients, for instance, in blood cancers, the biggest threat in terms of infections is TMV. And V delta 1s are the biggest protection, together with NK cells, I should say, against CMV reactivation. And that we know now, because when people are doing stem cell transplantation, if they spike the transplant with V delta 1 cells or with NK, provide protection against CMV reactivation. So there's a new protocol for stem cell transplantation that rather than taking out T cells to prevent graft versus host disease, they take out alpha beta T cells, which are the bad guys that create graft versus host, but they leave gamma delta cells in. And these gamma delta cells prevent CMV. We have papers showing as well this, and some unpublished data, that our dot cells are very good against CMV. They also produce a lot of factors that are interesting against HIV. And so this is another possible application of our cell therapy against virus. Against bacteria, no. They are terrible. They do not recognize bacteria. But other gamma delta cells, the so-called V delta 2, that we are not working with, but many other people are, they are excellent to recognize bacteria. They expand like crazy in TB patients, for instance. So we would say there are two arms of the gamma delta response in terms of infection: an antiviral, which is the one we are using against tumors. And you know, viruses and tumors share the aspect of being intracellularly manipulated cells, which is very different from bacteria, especially those that are extracellular, for which other gamma delta cells do the job.
Thank you very much, Bruno. So I didn't get to all the questions. I invite you, if possible, to go through the chat and answer people if you have a few minutes. And on behalf of everybody in Toronto and all of the other chapters, I would like to thank you again for joining us this morning for this wonderful talk. And
thank you very much.
Enjoy a great day in Lisbon. I'm going to turn it over to my colleague Ashton, who will introduce our next speaker.